Hello students, today's topic is molecular mechanisms of drug action. The mechanism of drug action occurs by means of physical interaction or biological interaction. For most drugs, the action is mediated through biological interactions, which could be receptor-based or non-receptor-based. Only a receptor-based interaction triggers a cascade of signal transduction. The receptors involved in drug action are of four types: the G protein based, the ion channel based, the enzyme linked, and the nuclear receptors. We had discussed about all these types of receptors in our earlier session. There are five basic mechanisms of transmembrane signaling. First, a lipid-soluble ligand that crosses the membrane acts as an intracellular receptor, as shown in the figure first. Next, a transmembrane receptor protein whose intracellular enzymatic activity is regulated by ligand that binds to it at a site on the protein's extracellular domain. That is figure 2. Third, the signal binds to the extracellular domain of a transmembrane receptor bound to a separate protein, tyrosine kinase, which activates. Figure 3. Fourth, a ligand-gated transmembrane ion channel that can induce to open or close by binding of a ligand. Figure 4. And last, the fifth one, a transmembrane receptor protein that stimulates GTP binding signal transducer protein G, which in turn modulates the production of an intracellular second messenger. In a present discussion, we'll discuss with examples only case 2 and 3. That is, a transmembrane receptor with intracellular enzymatic activity and the transmembrane receptor which stimulates an intracellular protein kinase. Both these transmembrane receptors come under the category of enzyme-linked receptors. There are three main types of enzyme-linked receptors. One, receptor tyrosine kinase or RTK. Second, serine threonine kinase. And third, tyrosine kinase associated receptors. In receptors tyrosine kinases, signal transduction occurs due to phosphorylation of the tyrosine residue at the cytoplasmic end of the receptor. Example, in case of insulin and EGFR receptors. We'll study both these pathways as examples for this. Next, serine threonine kinase. This specifically phosphorylates the hydroxyl side chain of serine or threonine amino acid residues. These kinases have a role in cell proliferation, differentiation, apoptosis, and even embryonic development. The next is a tyrosine kinase associated receptor where the cytoplasmic kinase domain is absent at the receptor end, but a cytoplasmic tyrosine kinase acts. Transmembrane enzyme-linked receptors. These receptors have two domains. The first domain facing the extracellular surface, which has the binding site for the ligand. Next, the intracellular domain, which faces the cytosol and has the catalytic domain, which could be a tyrosine kinase, serine kinase, or guanyl cyclase, as shown in the figure. Here, Y has the catalytic domain. This could be either intrinsic to the receptor or tightly bound to the transmembrane domain. The ligands that bind to these type of receptors are insulin, epidermal growth factors, platelet-derived growth factors, etc. Signaling mechanism in case of ligand-regulated transmembrane enzymes including receptor tyrosine kinases. These type of transmembrane receptors have tyrosine kinase at the catalytic domain facing the cytosolic end. These receptors are monomeric when inactive. When the ligand binds to these receptors, dimerization occurs between two neighboring receptors, leading to autophosphorylation. This activates the receptors and it leads to recruitment of certain proteins in the cytosol. The SH2 domain of the protein binds to these phosphorylated receptors and these proteins get phosphorylated. This activates other functional proteins which eventually results in stimulation of gene transcription or enzymes such as phospholipases and second messengers. As an example, we would take two cases, one EGF receptors and insulin receptors. The first example I would discuss for enzyme-linked receptor tyrosine kinase is signal transduction by means of growth factors. I request you all to go through the figure as I am explaining. The epidermal growth factor EGF binds to the EGFR receptor. The EGFR receptors are monomer units. When the ligand EGF binds to these receptors, it results in dimerization with neighboring receptor tyrosine kinases. The dimerized RTK phosphorylates one another at the tyrosine residues on the intracellular part of the receptor. This activates the signaling pathway. The dimerization and phosphorylation at the receptor leads to recognition site of adapter protein GRB2. The SH2 domain of the GRB2 protein binds to the phosphorylated tyrosine domain of the receptor. This activates the GRB2 protein. Next, this leads to recruitment of another protein SOS, which is a guanine nucleotide exchange factor. The SOS, when activated, is capable of replacing GDP to GTP. This is not shown in the figure. The SOS protein binds to the activated GRB2 at the SH3 domain. The SOS acts on inactive RAF protein, which is membrane bound, and inactive because it is bound to GDP. So the SOS acts on the inactive RAS protein and makes it active by converting GDP to GTP. The activated RAS protein activates RAF kinase, also known as MAPKKK. The RAF kinase, which is a serine threonine kinase, phosphorylates and activates MEK, that is mitogen activated protein kinase, also known as MAPKK. The activated MEK phosphorylates ERK, or also known as MAPK. The MAPK acts on DNA and regulate many transcriptional factors leading to altered gene regulation. The growth factors induce growth. Mutations of these could lead to oncogenesis. Another example that we would study under tyrosine kinase receptor is the insulin receptor. Binding of the insulin molecules activate the PI3K AKT pathway. Now, the insulin molecule binds to the alpha subunit of the receptor, which is a monomeric unit. Activation of the beta subunit at the cytoplasmic region leads to dimerization followed by autophosphorylation. This recruits a number of signaling molecules such as GRB2SOS RAS, leading to the MAPK pathway, the one we discussed for the growth factor receptor. Other than this, activation of insulin receptor leads to phosphorylation of insulin receptor substrate 1 and 2. IRS1 2. Phosphorylated IRS1 2 recruits P85 and PI3K, that is phosphatidylinositol 3 kinase. They bind to IRS1 and 2. This leads to activation of PI3K. Activated PI3K phosphorylates membrane-bound phosphatidylinositol PIP2 and PIP3. It increases the cellular level of PIP3 and PIP3 accumulates in the cell. Increased level of PIP recruits PDK1, that is phosphoinositide dependent kinase 1, and protein kinase B, also known as AKT, towards the cell membrane. PDK1 phosphorylates PKB and activates it. In the cell, reservoir membrane vesicles of GLUT4, that is glucose transporter type 4, are present. That is in case of muscle and adipose tissues. Translocation of GLUT4 vesicles are inhibited by the presence of an AKT substrate AS160 in the cell. In the presence of insulin, activation of PKB occurs, which inhibit AS160. Inactivation of AS160 leads to transportation of the transporter molecule GLUT4 vesicles to the cell membrane. This allows glucose to enter the cell and undergo glycolysis. Now coming to the next signaling mechanism, transmembrane receptor that binds and stimulates an intracellular protein tyrosine kinase. These are tyrosine kinases associated receptor families or non-receptor tyrosine kinases. These receptors do not have a tyrosine kinase domain, instead have a separate protein tyrosine kinase at the cytosolic end that binds covalently to the receptors and initiates the signal transduction. Example to these are cytokinin receptors. Cytokinin receptors make up the largest family of receptors that relay the signals by cytoplasmic tyrosine kinases. Examples of these type of receptors are growth hormone receptors and several kinds of interferons. Here, the signaling is transferred or signal transduction happens by JAK's STAT pathway. The basic mechanism of an nRTK pathway. As example, I have taken an interferon. When the ligand binds to the receptor, dimerization of two neighboring receptors occurs, making it an active dimeric state. The associated genus kinases are brought together. They go for autophosphorylation and also phosphorylate the tyrosine molecule or the domain. This activates a gene regulatory protein, STAT. The STATs, when phosphorylated, dissociate from the receptors and are transported to the nucleus where they modulate the transcription. You would understand it better with an example of a alpha interferon receptor. Example, alpha interferon receptors. When the interferon alpha binds to the specific monomeric receptor, dimerization of the two neighboring receptors occurs, as shown in the figure B. The associated JAK or genus kinases are brought together due to dimerization and cross-phosphorylation occurs. There are two different receptor chains associated with JAKs, that is TYK2 and JAK1. The JAKs phosphorylate the tyrosine residue on the receptor placed on the cytoplasmic site. The TYK2 and JAK1 recruit two different STAT molecules, STAT1 and STAT2, that is signal transducer and activator of transcription. They are phosphorylated by JAKs. Upon phosphorylation, the STAT molecules get activated and dimerize by attaching to one another's tyrosine phosphate and dissociate from the receptor, as seen in figure E. The STAT dimers translocate to the nucleus and binds to specific DNA sequences and regulate transcription, in this case leading to transcription of inflammatory mediators. This slide shows the differences between a RTK and a nRTK pathway. In today's session, I discussed signaling mechanisms of enzyme-linked receptors. We dealt with two enzyme-linked receptors, RTK receptors and nRTK receptors. We studied in detail the molecular mechanism of signaling for two RTK receptors, that is EGF receptor and insulin receptor, and for nRTK interferon alpha receptors. With this, we finish the signaling mechanism in case of enzyme linked receptors. Thank you.